Well, good morning. Thank you for joining us today. I'd like to start by acknowledging that the University of Manitoba campuses are located on the Treaty 1 territory of the original lands of the Anishinaabeg, Cree, Oji-Cree, Dakota, and Dene people, and on the homeland of the Métis Nation. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. Well, today's lecture is part of an institutional visit by the winner of the 2021 Henry G. Friesen International Prize in Health Research, Dr. Lorne Tyrrell. Dr. Henry Friesen, a proud Manitoban and outstanding clinician-scientist, is a legend for transforming academic medicine in Canada as he oversaw the transition of the Medical Research Council to the Canadian Institutes of Health Research. And Henry, we're glad to have you here today. I'd also like to thank the Friends of the Canadian Institute for Health Research, and in particular Dr. Obi Angel, the former chair of the Department of Internal Medicine here at the University of Manitoba, who's also here today for funding Dr. Tyrrell's visit. The Friends of CIHR created this prestigious prize in 2005 to recognize exceptional innovation by a visionary health leader of international stature. Dr. Tyrrell definitely meets this criteria. A distinguished professor at the University of Alberta, he's an innovative clinician-scientist, creative virologist, entrepreneur, and academic leader. Dr. Tyrrell earned his medical degree from the University of Alberta in 1968 and received his PhD from Queen's University in 1972. He completed internal medicine and subspecialty training in infectious disease at the University of Alberta and postdoctoral training in virology at the Karolinska Institute in Sweden. Dr. Tyrrell led the University of Alberta's Faculty of Medicine and Dentistry as Dean from 1994 to 2004. And nationally, Dr. Tyrrell was chair of the Association of Canadian Medical Colleges, where he rallied all the medical deans in Canada to support Dr. Friesen's vision of transitioning the Medical Research Council to the Canadian Institutes of Health Research. Dr. Tyrrell has held many important chairmanships, including the Institute for Health Economics, the Gardner Foundation Board, and membership of the Scientific Advisory Board of Health Canada and the National Vaccine Task Force. Dr. Tyrrell's most celebrated life-saving accomplishment in collaboration with Morris Robbins was the licensing of the first oral antiviral drug for chronic HBV, lamivudine, in 200 countries. This led to the creation of the Li Ka-shing Institute for Virology at the University of Alberta in 2010, and where he is founder and director of the institute. Dr. Tyrrell maintains active research programs in viral host interactions, development of HCV vaccine, and a program on SARS-CoV-2 vaccines, diagnostics, and antivirals. He's published more than 250 peer-reviewed papers, and Dr. Tyrrell has received many honors and distinctions throughout his career, including the Alberta Order of Excellence in 2000, Officer of the Order of Canada in 2002, and Fellow of the Royal Society of Canada in 2004. In 2011, he was inducted into the Canadian Medical Hall of Fame and was awarded the Killam Prize for Health Sciences in 2015. Please join me in welcoming Dr. Tyrrell.
Thank you very much. Well, thank you very much, Dr. Nickerson, and it really is a pleasure to be here today and to present the Henry Friesen Lecture and to have Henry here in the audience. So thank you. And there's such a history that I know in Manitoba of what I would say was academic medicine in Canada and how Manitoba has played a key role, particularly in our field with infectious disease. I just want to acknowledge Alan Ronald, who really is the father of infectious disease in Canada, and internationally, University of Manitoba is known for what they've done in this area. Also, Arnold, everyone knows his accomplishments, and Aubrey, thank you. Let me say that this lecture is a little bit different because it's going to be in two parts. I do want to talk with Aubrey of this. He said we should talk about your science, but also I wanted to talk a little bit about the vaccine task force and the pandemic, because this was ongoing at the time and I felt we needed to hear more. And I just want to say at the very beginning, the vaccine task force was such a unique experience. And I hope that you'll gather that at the end of the lecture. So in the first part, I'll talk about hepatitis B and account for some of our contributions to both hepatitis B and hepatitis C. And going from discovery, and it's important for students, I think now, that we do look more favorably upon translation and commercialization. There was a time in universities you didn't associate very much with industry. Today, we do. I tell the funny story of our university president who was questioned by a faculty member who said, "Don't you think the money coming from industry," and we had a contract for this, "is tainted money?" And he answered it very neatly. He said, "You know, you're absolutely right. It is tainted money. There tain't enough of it." And that, I think, is really echoes home today more. And then the second part, we'll do the vaccine task force. Let me say Henry Friesen, everybody here knows so very well, has contributed so much in Canada and to health research and health policy. He was the president of MRC from 91 to 99. He was the architect of going from MRC to CIHR that allowed us to dramatically increase our budgets. He would also recognize the very important position that Canada should be part of the Human Genome Project, and Henry was behind and founded the Genome Canada and served as the chair of that board for a period. He won an International Gairdner Award, which is the top award from the Gairdner Foundation for his work on prolactin, both in disease and in health. And he later won a Gairdner-Reitman Award, one of the few people to win 2 Gairdner Awards. He's the Royal Society Fellow, Companion of the Order of Canada, and in 2001 was made a member of the Canadian Medical Hall of Fame as well. Very distinguished, and I'm very pleased and humbled to be here to give this lecture today. Let me say that when I started practicing infectious diseases, I was doing a lot of classical infectious diseases, but I started to lean towards, I wanted to see more, and a brief story that I did look at my postdoc. Instead of going to Stockholm, I thought about going to the NIH and with Bob Purcell because I had a liking for hepatitis B. Actually, when I first went to university in medicine, I thought I was going to be a rural physician and practice in rural Alberta. But in second year medicine, I was fortunate to win an MD PhD scholarship that actually was based in New York, and it changed my career. So I had an opportunity to go away and train after this, and I thought of going with Bob Purcell. And in the discussion, he said, well, there's absolutely no way you can work in hepatitis B unless you've got a chimp colony close by, because you can't grow this virus in anything but chimps, and there's no cell culture system at the time. And so I did not go into hepatitis B in the beginning. But always I felt an urge to maintain some interest in hepatitis B and viral hepatitis. And as an infectious disease specialist, I always felt viral hepatitis should be part of our practices, as well as the hepatologists and the gastroenterologists. And I started seeing hepatitis in patients. Let me just say that in the world, on Tuesday, I don't know if many people know, but on Tuesday was the day that the world population hit 8 billion. I just saw that a little news flash that this had happened on that day. And about a third of the world's population has been infected with hepatitis B. And a number of those people don't get rid of the virus. They end up as chronic carriers. And most chronic carriers are susceptible to developing cirrhosis and liver cancer, 2 end-stage diseases. And there's about 300 million people in the world that are chronic carriers of hepatitis B. We have an excellent vaccine, and we can prevent it, primarily prevent the transmission from mother to infant, as all over the world that's still the number one way of it's being transmitted. But we can interrupt that with vaccines, but we don't do much for the chronic carriers. And I started to follow chronic carriers and was distressed to see their outcomes without any therapy. Let me say in the world's population, there's 3 major chronic diseases: hepatitis B, hepatitis C, which causes about 500,000 deaths a year, and HIV. I would point out these last two diseases we don't have vaccines for, but we do have really wonderful antiviral therapy, and those diseases have the potential to be well controlled or cured with antiviral therapy. In your life, some events in your life changed the course of your career. I was working on measles and MS, had CIHR funding, or pardon me, MRC funding at that time. But I gave a lecture in biochemistry in a graduate course in virology. Came across this paper that had just been published by Jesse Summers and Bill Mason, and read the paper and loved it. And I thought, this is a unique system, and maybe they use the duck model to work out the replication. I called Jesse Summers and said, Jesse, is there any way has anybody talked to you about using the duck model to find antivirals for humans? And he said, no. And I said, are you interested in working on that? He said, not really, I'm a basic scientist. I said, well, I would like to try to do this. And I sent a postdoc from my lab down to Fox Chase, and he brought back and we set up the primary duck hepatocyte system so we could have a duck model or a model of hepatitis B that might be a surrogate for the human. Let me just say the replication of hepatitis B is interesting in that the virus binds, it enters the cytoplasm, it rapidly uncodes and goes into the nucleus, and it forms covalently closed circular DNA. And covalently closed circular DNA has histones on it. It becomes decorated like chromosome, and it's like a mini chromosome in patients that are infected with hepatitis B, and it is the source for the transcription of messengers, and the virus is actually produced in the cytoplasm again. Some of the virus will go back in and build up the amount of cccDNA in hepatocytes, and the rest of it gets exported as secreted virus. Let me say our antivirals that we developed work out here in the cytoplasm. There's still a huge problem in trying to cure hepatitis B. We can treat them, but we can't cure them as easy as we'd like because of the cccDNA. Let me say that what was rather intriguing about this was that the viral polymerase for hepatitis B has almost the same qualities as the HIV polymerase. There is a reverse transcriptase, there's an RNase H, meaning it digests away the RNA in the presence of an RNA-DNA hybrid, so RNase H, and the DNA polymerase. It has one other key property, and that is a primer protein. The polymerase at the N-terminal end has a protein that is, or part of the protein is involved in priming the DNA synthesis. And so, unlike DNA synthesis in us that is not primed by proteins, this was a unique target that we thought might be a target for hepatitis B to inhibit the virus selectively. I would just say that at the time we started, there was still no cell culture for the human virus, only animal models, human chimps, but when the virus was found in ducks and the replication was worked out, we just asked the question, could this be a surrogate? My work would not have been possible without the association and collaboration with an outstanding nucleoside chemist by the name of Morris Robbins, who was at the University of Alberta. We went to the faculty club, as people do, and I drew out the replication cycle for hepatitis B and wondered if he was interested in working on this. He said he was from the very beginning, and he had some nucleoside analogs that we could begin to work with. Morris, by the way, was the first to synthesize DDG and DDC, DDG and DDA, the purine analogs, and Sanger, who got Nobel Prize for sequencing and the enzyme sequencing, got his supply of DDG and DDA from Morris Robbins. So he was an outstanding nucleoside chemist. I had no money when I started this. This is actually ducks on my parents' farm. And we raised the ducks out there. We'd go out and visit them, pick up duck eggs, come back, incubate them in the lab, and we started setting up primary cultures. Quickly across the top, the darker the dots, the more virus there is. And I would just say that at day 20 And the third dot in each of these is the comparison. And DDT had very little or no activity. The same thing true for DDC. But DDG, dideoxyguanosine, was more than 10,000, probably 100,000 times more active at that time. You just could not see any replication of hepatitis B in the systems we were looking at when DDG was added. So you wondered, why was this happening? Because you were incorporating dideoxys downstream, and we thought there must be proofreading, even though there's not the motifs for proofreading in the viral polymerase of hepatitis B. Somehow this was being proofread out. But you'll notice that the first nucleotide attached to the primer protein is always deoxyguanosine, and it's the same for all of the hepadnaviruses. So when DGG got bound to the primer protein, that bond could not be proofread out, whereas the others downstream could. And really, the DGTMP attached, and then you can prolong the chain elongation. However, when you put in the dideoxy, 100% of the DNA synthesis of hepatitis B was stopped. Skipping on to looking at a series of ducks. These were 12 ducks that were carrying hepatitis B. The first 4 were treated with placebo. The next 4 were treated with DDG, prodrug of DDG. I won't go into that, but it's a prodrug, and the last one, DDA. Just to show you that DDG was by far the most potent, and in a week we could see the virus was disappearing from the blood. In 2 weeks, 3 weeks. I really at that time thought maybe we have a cure for hepatitis B, which would be very nice. And we published this work, of course, on the comparison of the purines and the pyrimidines, and showing that this is very different. We then approached Glaxo and went down to Research Triangle Park and made the presentation. We had evidence in cell cultures. We had evidence in the animals. By the way, DDG did not show any toxicity in these animals. DDC showed some toxicity in others, but not the DDG. And presented to a group in Glaxo, and they said, just a minute, we're going to bring back, and they brought back the president, who was a Dr. Saunders from, it was Glaxo at that time, later became Glaxo Wellcome. And Dr. Jim Saunders came in and said, we like what you're doing, I hear, and we're going to work with you. And that other things that are important is Canada had just passed Bill C-22, and it meant that these companies were going to invest in money, some of the money of drugs sold in Canada back into research. And I actually did the research contract at an airport in Toronto, meeting with Jacques Lapointe, who was the president of Glaxo Canada at that time. Long story short, we did a lot of screening for Glaxo as well. Morris Robbins and I were at a Gordon conference, and we saw lamivudine having activity against HIV. And we came back and screened blindly compounds from Glaxo, and we asked them to include some lamivudine. Blindly did this, and we found that lamivudine was very active. It's synthesized in a plus enantiomer and a minus enantiomer. The plus enantiomer has toxicity. The minus enantiomer did not. This is related to mitochondrial toxicity. And the minus enantiomer was very active. We're a little bit surprised because this was a pyrimidine analog, not a purine. And it was synthesized by Bernard Boileau, and it had been licensed through Biochem Pharma to Glaxo. And then we requested it from Glaxo Wellcome at this time and tested it against the hepatitis B. Just let me say that, again, we'll turn back to the duck. And yes, there are 6 ducks. There were 3 of them treated with lamivudine, 3 with placebo. And you can see these are hours, at 8 hours, 16 hours, 24 hours, how rapidly the virus was turning over and how rapidly we could get rid of the virus in the serum. Again, thinking we had maybe a possible cure for hepatitis B. However, we've treated ducks for, as Alberta will know, he was in the lab at this time, Alberto Severini, who's here at NML, but we treated these ducks for a long time, sometimes up to 2 years. And when we stopped, disappointingly, when we stopped, the virus would come back. That comes back to that cccDNA, and any of the students that are out there, there's labs that are now looking at ways to get rid of the cccDNA, and we've continued to work on some of that. Of course, we did this in ducks. We didn't know if it was going to work in the human virus, but Ron Keeney, who was with us in Glaxo, had located a colony of chimpanzees that had been used by Bob Purcell, and Bob Purcell had infected a number of animals, and then they got placebo vaccines and challenged, and they ended up as chronic carriers. So we screened the new colony in New Iberia and found some young chimps that had been transmitted the virus from mother to infant. And these chimps were very well trained. They loved drinking Tang, and we simply spiked their Tang with lamivudine. I'll just show you that this little chimp started out with a very high level of hepatitis B DNA in the blood, and every 2 weeks we increased the dose, and then came back to the lab and to see if lamivudine had that effect on the human virus. And it was a wonderful day in the lab when we looked at this and plotted this out, showing how very sensitive the virus was to lamivudine. The mechanism, we were a little puzzled because this was now, why did lamivudine have this effect, which was a pyrimidine? Well, it does get incorporated into the virus, and where you normally incorporate cytidine, and but it didn't get proofread out. Now, the virus replicates inside of replicating cores inside of the cytoplasm. They're very small volume, but the DNA polymerase is reversible. If you have enough pyrophosphate, you can rechange this. And in the environment of the virus and the replicating cores, small enough volume, and you generate about 3,000 molecules of pyrophosphate through the synthesis of hepatitis B, and it tends to stay in the replicating core because of the lysines and the charge on the inside. But dideoxycytidine, dideoxyguanosine, incorporated downstream can be proofread out. But when you put in 3-TC or lamivudine, it could not be proofread out at all with the pyrophosphate. So it was unique in that it had a different mechanism, and it did not get proofread out by the phosphorylasis that did remove the others. So this one, we had a very good student working in the lab, and the mechanism of this was, again, you'll notice the first author in this, Alberto Severini, did a lot of work in the lab and how we looked at hepatitis B with the 3-TC and the mechanism. So thank you. Of course, this led quickly to a clinical trial, and the first clinical trial was done. I led that clinical trial with 5 different sites, and we put patients on lamivudine and showed that if they got as much as 100 milligrams, an aspirin is 375, it's like a third of an aspirin once a day of lamivudine. These patients showed completely more than a 99.9% decrease in their viral DNA. And this went into many different clinical trials, but the important one big one was in China, where patients were on placebo or lamivudine and showed a clear difference in the outcome with progression of the disease, and the development of liver cancer was decreased by the lamivudine. So this was licensed in 1998, and it was the first oral antiviral for hepatitis B. Let me say that lamivudine also, what was happening at this time is that people had stopped transplanting patients that had hepatitis B because they rapidly were reinfected immediately with the virus, and they would often develop a more aggressive disease we call fibrosing cholestatic hepatitis. Really, it's a very high level of the virus in the liver when there is no immune control, and it was really a fatal disease, most of them, and they'd have to be retransplanted again, and so they stopped transplanting many hepatitis B patients. We did put a patient on lamivudine, got ethics approval before it was actually licensed as a drug, but we started putting the patients on lamivudine pre and post-transplant. And the very first patient in the world that I think received an antiviral pre and post-transplant was done by Norm Kniedemann at the University of Alberta, May the 1st, 1994. Actually, we got the organ was flown up from San Francisco because this patient was end-stage liver disease, in ICU, in hepatorenal failure with the disease, and did very well. I should say did very well, but sometimes communication medicine isn't as good as it should be. He was on lamivudine for 9 years, and then he was in the transplant clinic, and somebody new saw him and said, why would anybody be on lamivudine this long? Stopped the lamivudine in April, and he came, I didn't know about this, and he came back into the hospital in fulminant hepatitis with B in July after it was stopped. But we do keep patients on suppressive therapy in the transplants, so they're done. Another nice thing about this was when we saw lamivudine being used is reversal of cirrhosis. This is a biopsy showing full-blown cirrhosis in a patient, 2.5 years on lamivudine, and the biopsies were back near normal. And today, about 70% of patients with cirrhosis that go on to the hepatitis B therapies will reverse their cirrhosis and not have to have transplants. So there's really no patient with advanced liver disease, if they got DNA, shouldn't be on antiviral therapy for Hep B. This was licensed in 1998. There was a problem, and we predicted that problem. Carl Fisher, who was actually in my lab at the time, came from Winnipeg as well. A number of people did, but Carl did some very nice work on this to show that mutations in the hepatitis B virus led to resistance, and we published a paper predicting the problem with lamivudine and resistance. When it was licensed, shortly thereafter, we started to see patients with resistance. Today, the drugs that are the best drugs for treating hepatitis B are entecavir and tenofovir. And these are purine analogs, and they do have that original mechanism of action as well as chain terminators. So I think the work we did early on the purines was a basis for some of this work as well. I want to touch the basis on a bit of work we did with hepatitis C as well. We began that about 1998 after we saw what the duck model meant. We wondered if we could get an animal model for hepatitis C. And there was only grown in chimpanzees and humans. We wanted to develop this animal model. That was done again in collaboration with Dr. Norm Kniedemann, a liver surgeon, and David Mercer, who was a postdoc, he was a graduate student after his residency in surgery. And he was putting human hepatocytes into skid beige mice, and they travel into the liver. We knew they went into the liver within an hour, and it would produce human albumin, but they would disappear. I came from a farm, and I remember in the discussion on his committee saying, you know, on the farm, if we don't spray the weeds, we don't get a good crop. And he said, what do you mean? I said, we need to somehow get rid of the mouse hepatocytes. There's room for the human hepatocytes to expand. And we went through several things we were going to try, but we ended up finding a paper that Brinster put out where they expressed urokinase behind the albumin promoter, and overexpression of urokinase destroyed the liver cells. This was for studies of bleeding disorders, but we adapted that and crossbred the animals carrying the transgene with our immune-compromised mice, and we produced mice that were immune-compromised, and when we put in the human cells, they would go into the liver and expand. We now can get mice produced that have 80 to 90% of their liver is humanized, and they were, remember how the albumin disappeared? In these mice, the albumin grew, and more transplant of human albumin appearing in these. And we infected these animals, and they would have concentrations of hepatitis C in the blood that was very similar to the human, and carrier levels, and they carried it for many weeks. So we had chronic infection of hepatitis C in a mouse model. We published that in Nature Medicine, and by the way, it was there were 4 different editorials in key journals that highlighted this as a key development for hepatitis research. The animal model has been used. This is the first mouse we ever had that had hepatitis C, and one of the things that was a side effect of this is chimpanzees in biomedical and behavioral research has really been stopped funded by the NIH based on the Institute of Medicine recommendation in 2010, and they quoted our work where we produced mice that had the xenotransplants that could be infected with hepatitis C as one of the reasons you don't need to continue to use chimps. Now, hepatitis B is the most common disease in China, and Mr. Li Kai-shing had been involved in Alberta. He owned a lot of husky oil, and we thought we had done something that helped the disease in China, and Mr. Li had made money in Alberta, and we approached him about one of his institutes. He's a philanthropist, and he's given money to 15 universities at least now that have Li Kai-shing institutes, and each of them are usually funded with a $25 million endowment. He came from Shantou in China. Shantou, if you don't know it, it's a little city in China about the size of Toronto, 4 million people. And recently he built a new medical school. When we first went there a few times, the medical school wasn't very great. The last time I went there, they had this new medical school, 9 stories high. You can see people in front of this. You can see it's a huge building, built by the same architect that did the bird's nest in the Olympics when the Olympics were in Beijing. Beautiful, very different but beautiful building, and has all the things, whistles and bells you can imagine. Manitoba has also been beneficial of Li Kai-shing. Money has come here as well. And they established the Li Kai-shing Institute at the University of Alberta. It was announced on April the 23rd, 2010. A good thing that was happening at that time, we had the CIRC program, Canada Excellence Research Chairs, and Michael Houghton, through our hepatitis C work, had been in contact, and we were able to recruit Michael Houghton to the University of Alberta on a Canada Excellence Research Chair. And Michael had met Mr. Li, and Mr. Li had stressed when we were doing this that he didn't want to see us just do research. He wanted to see research that actually benefited patients. And as a result of that, he stressed that, and Michael understood that very well and had worked in Chiron, discovered the hepatitis C virus, and worked on diagnostic tests for it as well. So we established not only the Li Kai-shing Institute of Virology, but the Applied Institute, so that any discoveries that we wanted to see commercialized, Michael was the perfect leader for that. And as you know, in 2020, Michael was awarded a Nobel Prize in medicine for his discovery of the hepatitis C virus. He led a team at Chiron to do this and worked very closely with Harvey Alter and Charlie Rice and others in making and confirming that he had actually found the virus. A little story on that is I nominated Michael for a Nobel Prize, or found people to nominate him for 8 years in a row. I just felt that that was an important discovery. Hep B had a Nobel Prize, HIV, and I thought hepatitis C would. On the day, the Nobel Prizes are always announced on the Monday, first Monday in October, and I used to get up and I'd wake up early in the morning, 4 o'clock in the morning, and check to see who won the Nobel Prize. And in 2020, I looked and there was nothing. And I looked about, as I was watching it, suddenly the announcement came out about the Nobel Prize, and that Michael Houghton had been one of the winners, which was a very memorable moment, of course. And they always call, the Nobel Committee always calls the winners, and that's the no. And so I waited about 20 minutes to make sure Michael had been called. And then I phoned him, and he said, I woke him up. And he said, Lorne, is everything okay? Calling in the middle of the night. I said, I'm calling to say congratulations. You just won the Nobel Prize. And the Nobel Committee didn't have his phone number, and later in the morning they phoned me for his phone number, but I had the pleasure of telling him first that he had won the Nobel Prize. So critical values, I think, to the success we've had in hepatitis has been, you have to really believe. And when I saw some of the results, and if you really believe that result is key, you've got to be passionate about it and keep persistent till you can carry it through. And I would have never done the things I've done without key partnerships and people that helped in so much, and so many very good students, a number of them from Winnipeg, and people who've come here. Let me move quickly on to the pandemic and say this COVID-19 pandemic is remarkable. We know that there's at least 6.5 million deaths, but it's probably closer to, some estimate, around 17 million deaths. Life expectancy for the first time has begun to decrease in some countries as a result of COVID. We've seen the stress on healthcare systems, not only in Canada but around the world, but Canada has seen major stresses. The economic effect, we're in trillions of dollars, and it's probably in the range of 15 to 17 trillion. And education has been set back 1 or 2 years with all of our closures. Societal effects with polarization of people within the same countries, polarization in families, deepened inequities, and there's many more examples you can think of. I just want to say that the death rate with COVID, when this is of 2021, was around 2%, and in August it was down 1%. And I just want to emphasize the benefits of vaccines. This just reflects what the vaccines have done in decreasing the death rates from COVID and how critical the vaccines have been, despite what the anti-vaxxers say and all the things that go on. There is an interesting publication that I'm sure many of you have seen, but it comes out from Johns Hopkins, and it said predicting a pandemic in 2018. This is now 2 years before the pandemic. They said it would be efficiently transmitted from human to human. It would have a non-trivial fatality rate. There would be no available medical countermeasures, vaccines or antivirals, immunologically naive population, so it's a new virus, capable of immune evasion, respiratory mode of transmission, transmissibility during the incubation period, and some percentage of the patients would be mild infection and transmit during that time. Couldn't have been more accurate in predicting what was going to happen. And when you look at the viruses, they were thinking, I think, at that time about influenza viruses, which is still a major threat. But you see in the red, infectious disease-related major threats in the world. In June of 2020, I was asked to serve on the vaccine task force by the federal government and the Ministries of Health and Innovation and Science. There were 12 people that sat on that committee. It was chaired by Joanne Langley from Dalhousie and Mark Lindeman from Toronto, who had worked previously in industry. But there were ex-official members that sat on that committee, and it included Ian Stewart, people you know here very well, Simon Kennedy, Stephen Lucas. And I want to highlight Anita Anand, who was the Minister of Procurement. She attended many of our meetings and reported where we were at in getting the vaccines into the country. We had an excellent secretary through Roger Scott Douglas and the NRC team. I've served on committees. This was different. We were advisory directly to the Minister of Health and to the Minister of Science, Innovation, Science, and Economic Development. And the deputy ministers are on all of our meetings. There were about 30 to 40 people from government that attended our meetings. We were told we needed to get vaccines for Canada that would be safe, effective, and timely. And we also were asked to help with the advice on rebuilding Canada's biomanufacturing and life sciences ecosystem. It was an onerous job. We met, in the beginning, we met often 2 or 3 times a week. And it was by Zoom. We never had any face-to-face meetings. They were all by Zoom, and the meetings ranged, the shortest meeting was 3 hours and as long as 6 hours. We met with manufacturers. We met with many different vaccine manufacturers, a lot of hours of Zoom, and we wrote 52 letters of advice to the government. What was different about serving on a vaccine task force to most committees? Usually committees get together and you write a report, and that report goes to government. It sits on a shelf and it may gather dust and never be acted on. This committee, every time the government was waiting for the letters of advice. So it really gave you such a lot of energy to work on this committee because it was direct to government and direct, you were knowing that within days, if not a week, a lot of your advice was going to be acted on. There was an urgency of the pandemic for the government and the urgency for us. I can tell you the first couple of months, I kept waking up at 2 in the morning and worrying about, are we making the right decisions? And I can remember my wife saying to me one day, she said, you know, you're not going to be much use of this. You keep getting up in the night and worrying about it. How do you work on it the next day? And, you know, you got to settle down. It was just very motivating. And as I said, the advice was waited for and acted on rapidly. We had weekly feedback. As we went from one meeting to another, Anita and Nan or her team would report where we are with procurements, and you knew how important that was for Canada. It may not seem it now, but at the time it was really... Public health measures, of course, are the first things that you want to look at, and we've seen with SARS in 2003, which NML and University of Manitoba had major effects on that. But you know, this had a mortality rate of 9%. And MERS has a mortality rate of 34%. And closure and public health events, we controlled the earlier ones, but couldn't control this one. And there were antivirals and studies. Over the time we were on this, there were a lot of antivirals to look at and a lot of vaccine candidates. And we knew that vaccines could be produced by different methods, but we didn't know how effective they would be. There were inactivated vaccines, live attenuated vaccines, vector delivery, adenoviruses, mRNA vaccines, self-applicating RNA vaccines, protein subunits. And so we made recommendations, and we knew that we had to make recommendations on a portfolio of vaccines. So we covered a fairly broad spectrum. Let me just first say that it was remarkable because most vaccines took 10 years to get into patients. At this time, what was happening so quickly, the first case was diagnosed on December 8th, 2019, and you all know the story. The first licensing of a vaccine was right on a year, on December 9th, 2020, we had the first vaccines approved in Canada. And December the 23rd, the Moderna, first was the Pfizer. Remarkable fast. And of course, the key people were Drew Weissman and Katalina Karako, and if you watched the Gairdner Awards this year, they were presented Gairdner International Awards, but Katrina's discovery that by modifying the uridine in the RNA could prevent the innate immune response. So putting pseudouridine in the place of uridine, normally when they tried to put mRNA into cells, you trigger an innate immune response and destroy the RNA. But putting a pseudouridine in place of the uridine, that didn't happen. So suddenly you could put mRNA into cells that would not cause an innate immune response and could be translated into protein. She did that in 2005, by the way, never recognized the importance of this and moved from lab to lab as a research associate. And settled down with Drew Weissman, but she left there and went to Germany with biotech, and then Pfizer partnered with biotech, but critically discovers key Canadian elements. And I just want to stress that there were key Canadian elements that we recognized in the vaccine task force, people who made the delivery of these RNAs into the cells. And Peter Cullis at UBC, as well as Ian McLaughlin at Techmira, did the key early discoveries on how you could modify the lipid nanoparticles to get these into the cells. But a very important Canadian contribution here, and I'm sure we'll see another Nobel Prize coming to a Canadian related to this. Of course, I'll just skip very quickly to this one, and these started Moderna, was started by two people who trained in Canada. Lou Barr was a postdoc at McGill and went to MIT, and Derek Rossi, who was a master's student with, or PhD student with Alan Bernstein, ended up going to MIT, and the two of them are really the people that started Moderna as this. They got advice on some of this work with RNA from a key Canadian that has had a lot of input into this, and that's Nachum Sonnenberg from McGill University, one of the world's leading RNA biochemists. We were hoping to see a 70% protection, like influenza vaccines, maybe. We were delighted when the results came out, and we had recommended these mRNA vaccines, and to see a 95% protection, both for Moderna and for Pfizer. And it gave us great relief. Let me say the other vaccines, we did recommend 2 that uses adenoviruses, both the AstraZeneca and the Johnson & Johnson or Jensen. And there's a key Canadian component to this. Frank Graham, who was funded by MRC for many years and was at McMaster University, worked on the delivery of RNA or proteins using adenoviruses that were modified, and his technology is what AstraZeneca and Jensen used. He also came up with the HEC293 cells, and many of us in the room have used HEC293 cells, and the companies that produce these vaccines have used HEC293 cells. And I just wonder if anybody knows, what does HEC293 mean? Why does it have that number, 293? Well, the real reason is it took 293 passages by Frank Graham in his lab to come up with the characteristics he wanted to see in those cells that allowed this to happen. And that's why Frank Graham is currently retired in Italy with his wife, and is enjoying some of the fruits of this work. The vaccine task force can be criticized. We've recommended too many vaccines. We hear that some of them are going to expire. We were told, make your recommendations with no regret. You don't need to worry. We need to get the vaccines and do what you think is right for Canadians. And that's what we did, and we did recommend 7 different vaccines. 5 of them were eventually approved in Canada, but the 2 that have carried the weight primarily have been the 2 mRNA vaccines. Just want to show you that this is work from another person who was from NML, who actually is at the head of the provincial lab, Graham Tipples, who's well known here in Manitoba. But it shows how dramatically one strain of COVID is replaced by another. This is the Wuhan in Alberta, and then we get to the Beta, the Gammas, and we go on to the Deltas, and then to the Omicron. It is remarkable how these viruses replace one another, the strains. And that's why you're going to see probably this virus is around and going to be endogenous for a long time. And particularly the Omicron, because just recently in Nature they came out and showed that the variants of Omicron have continued on more than the other viruses. And we're now out here with the BA4 and BA5s, and that's what you were seeing here is the BA4 and 5 in Alberta. And this is a picture of what's happening across the world. If you look at it, the R value shows how readily they are transmitted. The Wuhan, 3.2, Alpha, 4.6, Delta, 5.9, and the Omicron's up there close to what measles is at 9 or 10 for their R values. There has been definitely changes. And the vaccines do not give you protection against infection 100%, and Omicron is clearly infecting people who are fully vaccinated. But the vaccines have changed the immune system in the person so that you get a better outcome than if you're not vaccinated. And I think we'll see more of that in vaccines. Vaccine administered in Canada, this is as of August. I should have updated that a little more, but I would really be concerned with what I call vaccine fatigue. And we do need to continue with the boosters. You need to continue to keep your immunization up. Otherwise, we're going to get into trouble with this disease going forward. Antivirals for COVID, I've been disappointed to see how much work was going on and how few actually were licensed, and some of them aren't that good. But I believe that new technologies, the protein structures that we can get from computational and artificial intelligence, the cryo-EM, and now using computational systems for drug design, we will see new antivirals coming in the future. This is the AlphaFold, and this is the how quickly both Meta and AlphaFold are giving us structures of proteins that we never believed, 600 million proteins. In the United States, they've looked at the future of pandemics and how you're going to present it, and they've set up key centers to do vaccines development for the key viruses they think will be the ones that will cause pandemics in the future. And they've set up 9 sites to look at antiviral drug development. And these are cooperative between major universities, including some Canadian universities. Matthias Gote from our lab from the University of Alberta has cloned all of the main polymerases of all the viruses of threat. I mean, the new virus that comes out, and within 3 weeks he's got it cloned and expressed. And he's part of some of these centers, and we will be able to find antivirals in the future. We have to keep funding pandemic preparedness. We've seen a Canadian just do this in Australia. We wish it had done back in, he's from Calgary originally. The Canadian government has set up a biomanufacturing life sciences strategy. They're investing a lot of money again, and it's in the CBRIF that we were talking about this morning, and the Biosciences Research Infrastructure Fund that they just announced, getting money into a number of universities to look at how we go forward with dealing with these organisms. Our committee made a number of recommendations, actually $1.2 billion of recommendations that have been acted on trying to rebuild the infrastructure in Canada. Some of this is in Manitoba, some of it's across the country. Pandemic threats in the future, as I said, SARS is 1% to 2%, COVID-1, 9%, MERS, 34%, bird flu, 50 to 60%. And bird flu has come back this year stronger than ever. My message at the end is the same thing as the World Health Organization said. Message for change is clear. No more pandemics. We've got to prepare properly. If we fail to take this goal seriously, we'll condemn the world to more successive disasters, catastrophic disasters. And I believe, traveling and spending this time, that pandemic preparedness has to have the same urgency as climate change. Because you can imagine, how would we survive as societies and worlds if we have another pandemic within 5 years instead of 100 years? It would have huge impacts, and we've got to be prepared for that. And we've got to have governments understand that, industry understand that, and we do have some great philanthropists that do understand it. The Gates and others, including Cummings. I'll stop there, but I just wanted to say that I learned from the Vaccine Task Force. I hope that committees in the future can work directly with government when they ask you to do, and you get this rapid response. It was very, very enlightening and exciting to be on it. And we need to see it continue. Thank you.
Thanks very much, Lorne. We have time for some questions, if there's any questions from the audience. Go ahead.
So thanks, Lorne, for the talk. One of the questions I have is back to this idea that I think you hit on at the end, is this idea of pandemic preparedness, but more so from a pandemic containment standpoint, and whether or not we can actually truly contain the next pandemic before it becomes a pandemic. One of the things that I think is critical in that is the investment, not only here and in high-income countries, but more so in the global south, where we know hotspots exist for emerging pathogens. Do you think that there will be enough of a concerted investment by the high-income countries of the world to truly be at a point of true containment of pandemics, or is it more going to be the fact that we will be prepared for when the pandemics occur because we simply won't invest in those regions?
Well, I think we've seen some investment in those nations and some patent releases in those nations so that they could prepare some of the vaccines, and we're looking at antivirals in low and middle income nations because, of course, pandemics are global and you need to be able to protect them as well as us. I think there are a lot of lessons that came out of this pandemic, and we need to do those things better. You know, I would say that there was a vaccine developed for SARS-CoV-1, and Michael Houghton was involved in that, and he supplied his vaccine to the NIH, and it was never developed for it because the disease disappeared. But I can tell you that we have serum from those, and there's a lot of cross-protection. Had that vaccine been developed, or others that were developed for SARS-CoV-1, we might have had much better outcomes in SARS-CoV-2. So I think that there has to be ways that governments, industry, people work together to make some of these tools that we can stockpile for the event that one of these viruses comes in, and we can have a much better head start and try to prevent it from becoming a pandemic. And I think that's where we got to go with some pandemic preparedness in my mind. There's lots of work that the social sciences have a huge amount of work to do since what has happened here. I mean, the anti-vaxxers, the misinformation that has had a big effect on how we deal with this and how governments are dealing with it. My government in the province of Alberta is a perfect example. And we're going through some difficult changes as a result of this misinformation. So there's so many different aspects that could be looked at, but there's a tremendous opportunity from what we've learned from this to try to be prepared better for the next pandemic.
Go ahead. Yeah, Ray.
Andrew. Okay, sorry, yeah, I wasn't sure you were pointing at you. Thank you, awesome talk. Similar vein of questioning. So, more so on the global political scale though. So there's the financial considerations which are important to take into account. But my concern is the sort of increase in divisiveness, I suppose, that has been rising in recent times, and then just seeing global powers working together to kind of, for example, China and the West, et cetera, however you want to paint it. Just having that communication, that back and forth to actually prevent a pandemic before it happens. I'm just worried about that. Like, how would you strategize, I guess, or plan about tackling that or diffusing that, if you will, going forward?
Well, I think there has been thoughts about this and how we do this, and the COVAX was set up to do this. Canada was part of COVAX, and we did, we ordered vaccines, but we've contributed about 200 million doses of the vaccines through COVAX. We're at about 140 million, and I think we're going to reach the 200 million that gets out to vaccination. But we need to do that much better. We need to be able to, if you're going to stop a pandemic, and it's found in countries, they often start in countries, they're often zoonotic, they start in areas of the world, and the NML and the Public Health Agency of Canada had a very good early warning system, and unfortunately that was wound down just before this, and it was lots in the paper, Globe and Mail, etc. But it's been reestablished, and it's really important that we had that. I think it was one of the best early warning systems in the world that was here at that time. And that has to be such an important part of pandemic preparedness, is the monitoring, the epidemiology, the diagnostic tests, and all of those aspects. And we have to be sharing. We have to find a way to prepare this on a global scale, not just for here. But Canada can, if we reestablish the biomanufacturing, we can be part of that on a global scale.
Well, I want to thank you, Lorne. I think it was a great illustration of how academic medicine within the universities and discovery science really has had an impact, and especially Canada, as you illustrated, has had an impact in bringing this forward, and how that basic science is absolutely critical to population science at the end of the day. And we started to keep talking about one or the other instead of the and of bringing them both together. And I think that this was a great illustration through the pandemic of how actually basic science informed population science. And we have to continue to build these bridges between the two.
Absolutely.
All right. Well, thank you very much.
Thank you.